And that's because carbohydrates are a primary source of energy. And carbohydrates, or glucose, is the most common carbohydrate we get energy from. So actually, we're only going to talk glucose catabolism. And if you understand glucose catabolism, you're going to understand the rest of the catabolism because they all use quite similar pathways, similar processes. Now, glucose is broken down in two general ways called cellular respiration. That's glucose and oxygen is used to make carbon dioxide and energy, with water as a waste, or fermentation. That's glucose is used to produce energy and some sort of organic end product. Now, these are the sort of big picture. Ignore some of the words on the side. You can read them if you like to help provide context, but I'd like you guys to kind of look at these big pictures. This is respiration on the left side. This is fermentation on the right side. And if you want to zoom in, you can even look at what I like to call sometimes medium-sized pictures. Right? These large arrows represent many, many reactions, many pathways. So we're going to look at each one of these separately as we talk about first respiration, and then fermentation. Respiration is also called the complete oxidation or catabolism of glucose. And what that means is that our end product, our waste products, are inorganic. Right? We're producing an inorganic end product. Most of the energy, i.e. ATP, produced is via oxidative phosphorylation, right? The idea of building the dam. And keep the dam in mind. Just think about as we're going through this whole process, we are simply building a dam. Right? So when you think to yourself, why are we doing all this? The answer is we're building a dam. Two types of respiration. Aerobic respiration is with oxygen. Anaerobic respiration is without oxygen. Right? But more or less the pathways are quite similar, and I'll show you a little bit where things may diverge, just a tiny little bit. And in cell respiration, we're talking a gigantic process. There are multiple pathways involved. Glycolysis, Krebs cycle, and the electron transport chain. These are kind of our medium-sized pictures, right? So look at each one of these, and we're going to look at sort of the big picture of the medium-sized picture, if that makes any sense. We'll start with glycolysis. Now, glycolysis is the oxidation of glucose. Right? So when I say oxidation of glucose, what are you thinking? Yes, you're thinking of removing electrons from glucose. And glucose is a 6-carbon molecule. At the end of the day, we're going to have 2 3-carbon molecules called pyruvic acid, sometimes called pyruvate, by the way. Right, so essentially what we're going to do is we're going to take glucose, 6 carbons, let's draw that in here, 1, 2, 3, 4, 5, 6, and we're going to split it in half and produce 2 pyruvic acids, or 2 pyruvates. We also have 2 main end products called NADH and ATP. NADH is an electron carrier. ATP is ATP. And ATP is produced via substrate level phosphorylation in this case. Right? So it's that quick production of energy. So now you should take a moment to use this arrow and fill in the gaps. Right? Fill in the blanks. So you can pause now. Write it in. All right, good job writing it in. So we start with glucose. We end with pyruvic acid. And now we've made 2 ATP. And you can write a little note via substrate level phosphorylation. Very quick, very easy, not super efficient. And NADH. And if you want to put some numbers, there's 2, there's 2. And there's 2, right? And that's sort of your big picture of glucose, right? And at the end of the day, you can say in glycolysis, we produced a little bit of ATP, some NADH, and we've split glucose in half. All right. So let's look at this strange molecule we call NADH. Now, NADH actually starts off life as NAD+, and NAD+ is an electron carrier. It's an empty electron carrier. And what do you think it does? It carries electrons, right? It has pockets where it can carry electrons. NAD+ can remove electrons from food, i.e. glucose, and when it does, it becomes NADH, right? So which one is oxidized? Which one is reduced? NAD plus is oxidized, NADH is reduced, right? Notice NAD plus oxidizes glucose, takes electrons, and itself then becomes reduced to NADH. I know, crazy stuff, right? So diagram that out, make sure that it makes sense to you. NADH now takes electrons away. I like to think of it kind of as a taxi cab, right? NAD plus is your vacant taxi cab, goes to glucose, it oxidizes glucose, becomes reduced to become your full or occupied taxi cab, and then it takes the electrons to their final destination. And we won't worry about that destination just yet. This pathway shows the nitty-gritty details of glycolysis. You do not need to memorize this for my class. One day, maybe in organic or biochemistry, you're gonna have to. I know I had to, but you will not in this class. So please do not stare at this and try to memorize the details. We're just gonna use this as the big picture type of look. I'd rather you look at this and just try to understand what's happening in glycolysis. What role does glycolysis play in the whole metabolic process? So a couple of things I want to point out. Requires a couple of ATP to get started, i.e. light the fire, right? So we actually have to input a little bit of ATP at first. And then along the way, we have NAD plus is reduced to NADH, right? Because it's reduced because it oxidizes something. What is it oxidizing? And if you said glucose, that is correct. And then at the end of the day, we're also making some ATP, right? 4 overall. But because we have to use those 2 up top, we have net overall production of 2 ATP in glycolysis. And then we have our final product, 2 pyruvic acids or pyruvates, and those are going to go on to the next pathway, the next medium-sized picture. Now, one thing that's kind of important, glycolysis does not require oxygen. Oxygen is not involved in glycolysis. It doesn't help it, it doesn't hinder it. You can do this in the air, in the absence of oxygen. You can do this with no oxygen around. You can do it with. Glycolysis does not care. And that's kind of important later too. There are other alternatives to glycolysis. It's not the only way that we break glucose, called the pentose phosphate pathway and the Entner-Duderov pathway. Just write this down, and we're probably never going to talk about it again. All right, so that was glycolysis, right? Our big picture here. Now we have pyruvic acid, and we're going to move on to our second, right over here, called the Krebs cycle. But notice we have this thing called acetyl-CoA. Right? So before we get to the Krebs cycle, we actually have something called the intermediate step. Some people call it the prep step. And this is where pyruvic acid gets converted to this thing called acetyl-CoA, kind of getting it ready for our next step. But along the way, a couple things happen. First, we make some more NADH, right? NAD plus, what is it doing to pyruvic acid? If you said oxidized pyruvic acid, that is correct. And then in itself, it becomes, if you said reduced, that is correct. Reduced to become NADH. Remember our full electron carrier. This guy is going to go off, and we're going to see him again soon. Oh, by the way, whenever we say NAD plus oxidizes something, you don't have to say exactly pyruvic acid or glucose 6-phosphate or whatever. Always just think, really, pyruvic acid came from glucose, so... Oh, I totally spelled that wrong. Glucose, whatever, you know what I mean, right? NAD plus oxidizes glucose, and that's kind of what we say. We also lose a carbon, right? So here we have 3 carbons, acetyl-CoA only has 2, so actually we lose a carbon, and this carbon becomes carbon dioxide. This is now waste, right? So we've generated our first waste product, yeah. And then we produce acetyl-CoA, which goes on to our next step. And our next step is called the Krebs cycle, sometimes also called the tricarboxylic acid cycle, sometimes called the citric acid cycle. Not too important what you call it, just understand that it's a series of 8 chemical reactions. And this chemical reaction, essentially last product, is the same as the first reactant. It's the cycle. During the Krebs cycle, we are now completely catabolizing our organic molecules. So all our carbons now are going to leave as carbon dioxide waste product. So after the Krebs cycle, we do not have organic glucose anymore. It is gone. The most important products of the Krebs cycle are these guys, NADH and FADH2. And FADH2 is also just simply an electron carrier. And so FADH2 is an electron carrier, and we also make 1 ATP via SLP. And these guys are going to go off. We're going to see them very soon. ATP is ATP, and second verse, same as the first, right? Looks like this, and again, no details, please. So I don't have to say that anymore. Here's our acetyl-CoA, enters the Krebs cycle, and then now we're going to go around the chute, right? Around the horn, and just like that. Along the way, some important things to note. Lots of NADHs and FADH2, right? We are continuing to oxidize glucose, right? We're oxidizing glucose, which means we're taking the electrons. NAD plus gets reduced. NADH, FADH2, they go off to do what they need to do. We will see them very soon, actually. We produce 1 ATP. This is via substrate level phosphorylation, direct transfer of phosphate to ATP. And we have generated carbon dioxide as waste, right? So that's now a waste product. We've now completely oxidized our carbon. So if you're keeping score at home, that's now 6 carbon dioxides produced, 1 for every carbon in glucose. And finally, we have our last product, same as starting reactant, right? And now that can keep going, keep the pathway going, provided that we keep on receiving acetyl-CoAs. And we will keep on receiving acetyl-CoAs if we keep on eating food. All right, so we have now talked about 1, talked about 2, and again, big picture, right? I really like these diagrams. You see these nice big pictures, carbon dioxide is leaving, produce an ATP, and you should say to yourself, oh, this ATP was produced by a substrate level phosphorylation, right? And then we have our NADHs and FADH2s. Notice where they're all shuttling to now. NADH from glycolysis, NADH, FADH2, all moving to our third and final major pathway called the electron transport chain. And notice this other word, chemiosmosis. Right? Remember we talked about that? That was that gradient we built, that dam. Now the electron transport chain is a series of electron carriers, electron carrier enzymes in the membrane. And for bacteria, it happens at the plasma membrane. For eukaryotic cells, it happens at the mitochondrial membrane. And they pretty much do what the name implies. They transport electrons in a chain. NADH, FADH2, they pass electrons to the electron transport chain, so they're giving up those electrons. So what is happening to NADH and FADH2 if they give up their electrons? You said they are becoming oxidized. You are correct. So they are becoming oxidized because they're losing those electrons. The electron transport chain enzymes are getting reduced because they're gaining. After NADH, FADH2 become oxidized. They go back to becoming NAD+, FAD. They return to glycolysis. They return to the Krebs cycle to pick up some more electrons, so then they can bring them back to the electron transport chain. And that's what they do. They shuttle electrons, like a taxi cab, back and forth. Now the energy that's in those high energy electrons that we're pushing around on our electron transport chain is used to take protons, H+, from the inside of the cell to the outside of the cell. So we're pumping protons out, and this concentration gradient is called our chemiosmotic gradient. Right, so we're doing the work in the electrons. The energy in the electrons is doing work to produce this chemiosmotic gradient. And at the end of the chain, the electrons end up on the final electron carrier, the last thing to carry the electrons that began in glucose. And then that final electron carrier becomes waste. And since that final electron carrier is receiving the electrons, what is happening to it? It is getting reduced. Great stuff. All right, whoa, look at this diagram, right? So we got this crazy diagram. Let's look at some of the particulars here. And I've blacked out this because I don't want that to be distracting. Here's NADH. Here's FADH2. Let's all pretend it starts at the same place. NADH becomes oxidized. It's giving up its electrons. Becomes NAD+. Now that it's NAD+, it's going to go back and go to glycolysis, go to Krebs cycle, and pick up some more electrons. These electrons now are getting pushed from one carrier to the next carrier, moving around just this way between all these different enzymes, and every time the enzymes or the electrons are being carried, protons are being pumped out. So they're being pushed from one side of the membrane to the other side of the membrane. What type of transport is this, by the way? We're talking about moving electron or molecules from an area of low concentration, because we're not going to have a lot of protons here anymore, to an area of high concentration. Notice how as we keep pumping protons out, we're going to have high concentration of protons, right? Active transport. Remember we talked about that? Right? Active transport, using energy to move molecules from an area of low energy or low concentration to high concentration. Now, we talked about normally ATP drives this. In this case, the energy used to drive active transport actually comes from the electrons from glucose, right? So when we're eating food, all that energy in food is used to create this chemiosmotic gradient. High concentrations outside, low concentrations inside. Now these electrons finally need to go somewhere. We can't leave that electron there. If we leave that electron there, it gets backed up, and the whole electron chain gets backed up, and then you can't continue to make ATP anymore. So we have our final electron carrier, oxygen. Oxygen picks up that electron, so oxygen becomes reduced, right? Reduction is gain. We are oxygen reducers, by the way. And when oxygen becomes reduced, it becomes water, which is our final waste product. Now, if you add up our equation, glucose plus oxygen becomes carbon dioxide, water, and energy. There you go. Although actually we have not yet made energy. All right, so let's look at our chemiosmotic gradient, H plus protons, right? We have this gradient. Now, where do these protons want to go? That's right, they want to go in the cell, right? Because our gradient always favors diffusion, high to low, always wants to go in. But can they? That's right, they can't, right? Because they're ions. Ions cannot cross that membrane. So let's look at what we've just done. Our electron transport chain, all the energy in the glucose that we've eaten generates this chemiosmotic gradient. Now, this gradient wants to flow into the cell, but cannot. But we can let it. And we can do it via facilitated diffusion. Remember, allowing diffusion through a transport protein. And our transport protein here is called ATP synthase. Now let's look at the name of this, ATP synthase. Right? We know that's an enzyme because it ends in ASE. And it synthesizes something. What do you think it synthesizes? ATP, that's right. So ATP synthase is actually able to capture the energy in that chemiosmotic gradient and produce ATP. And this is oxidative phosphorylation. Finally, we have made ATP, not by directly transferring phosphate to ADP, but by building this gigantic dam, our chemiosmotic gradient, allowing that gradient to go through this ATP synthase, driving our turbine, and finally producing ATP. It looks something like this, right? So very simplified. We have our electrons going through our electron transport chain, pumping out our protons, high proton concentration, low proton concentration, wants to flow into the cell. We allow it to come through this ATP synthase, and every time it comes through, these turbines spin, and then allowing this phosphorylation of ADP to make ATP. Right? And that is oxidative phosphorylation. All that work, all that stripping of electrons, and you're like, why are we even making this NADH? And why do we keep oxidizing glucose? Why don't we just burn it in glycolysis to make all our ATP? Well, this is why. Creating this gradient, making this chemiosmotic gradient to produce ATP. Now, is it worth it? Well, let me give you some numbers here. I think I might have them later again, but we'll just see. 4 ATP produced via substrate level phosphorylation. 2 here, 2 there. That's 4 ATPs per glucose produced by substrate level phosphorylation. Oxidative phosphorylation produces 30 to 32 ATP from that same glucose. So look how much more energy we get. Right? A lot more energy, but it took a lot of work to do it, didn't it? Right? It's a whole bunch of energy. And that kind of becomes significant later, as I'll discuss. Whew. So, our overview of respiration, this is like a gigantic picture. Hopefully now this starts to make a little bit more sense. And try to look at it in a series of medium-sized pictures. Again, try to understand what happens at each step, what goes in, what comes out, what are some of the key features. Don't get lost in the tiny little details, right? We don't want those tiny details. Very quick overview or summary of respiration. Two types of respiration. Aerobic respiration is all about using molecular oxygen as our final electron acceptor, and we call our organism an aerobe. Anaerobic respiration is done by anaerobes, and it's using a final electron acceptor that is not oxygen. It's some sort of inorganic ion, and it can involve things like nitrate ion, sulfate ion, carbonate ion, right? So various inorganic ions. And overall, it yields a little bit less energy than aerobic respiration because some of the Krebs cycle doesn't work quite as well under anaerobic conditions, right? So it yields a little bit less energy, not a huge amount. We're talking maybe 34 for aerobic respiration versus 26 to 28 for anaerobic respiration, maybe as little as 20, depending on what you're using as an electron acceptor. But, still less. So overall, aerobes grow faster than anaerobes because they're able to produce more ATP per glucose. Here is a very quick score sheet of where our energy comes from. Again, notice 4 from substrate level phosphorylation. We can get up to 34 from oxidative phosphorylation, right? So huge amounts of energy. Maybe takes a lot of work, but huge amount of energy. Now, of course, glucose is not the only thing that we get energy from. There are other energy sources. But I don't want to get into all the different pathways because, as you can see, most of the pathways involved all funnel into our glycolysis Krebs cycle electron transport chain, right? So we can, the basic difference of the pathways are simply to funnel them, convert them into products that can then feed into glycolysis, Krebs cycle, and ultimately the electron transfer chain, right? So the machinery isn't all that much different. All right, so that was respiration. Now let's look at fermentation. Very important, probably my favorite metabolic process because it does so much good for us. And there are a lot of different definitions for fermentation. Some people talk about it as a spoilage of food, right? So when microbes spoil food, that's a fermentation. Any process that produces alcohol or dairy products, right? The good stuff, that's fermentation. That's my favorite type of fermentation. It produces our beers and wine, yogurts and cheeses and things like that. And any large-scale microbial process uses fermentation, or it's called a fermentation. Now, typically fermentation does not involve oxygen, but the large-scale process can involve oxygen, right? So that's called fermentation or industrial fermentation. We're going to look at it from more of a scientific definition, right? The literal definition of fermentation. And it's all about releasing energy from the oxidation of organic molecules, right? Again, here's that word, oxidation, or stripping electrons from organic molecules. Does not require oxygen, and the reason for that is because it does not use the Krebs cycle or the electron transport chain. Right? So it doesn't use oxygen, is not harmed by no oxygen around, doesn't gain when there's no oxygen around. So maybe a better word here is that it's indifferent to oxygen. It really doesn't care whether oxygen is present or not. And the organic molecule, the final electron acceptor is an organic molecule. And the waste product ends up being an organic molecule. So there's still energy left in the waste product. And overall, you really don't get a lot of energy from fermentation. But they have their own ways to compete. In fermentation, ATP is generated only via glycolysis. Right? So in fermentation, we start with glycolysis just like before. The second step of fermentation is converting pyruvic acid into an organic molecule and product. Basically, the pyruvic acid is being used as a final electron acceptor So when pyruvic acid gets reduced, right, or those electrons get dumped on it, it then gets converted into the waste product. And overall, a small amount of energy. Now let's look at why this even happens. Right? So here we have glycolysis. Glycolysis goes to pyruvic acid. We've made our ATP. This goes on to do some sort of cell work, and then eventually goes back and becomes ADP again, right? This is kind of our limiting factor. NAD plus reduces glucose, oxidizes glucose, becomes NADH. Normally, in respiration, NADH goes off to the electron transport chain, remember? And over there, it becomes NAD plus, and then it comes back. So we can complete our cycle. But since fermenters, they don't use the electron transport chain, it can't restore its own NAD plus or NADH. So in this case now, we're going to require, we need to regenerate our NAD plus, right? So this NADH that's produced during glycolysis needs to be regenerated. So NADH reduces pyruvic acid. So our pyruvic acid gets reduced, right? It gains those electrons. NADH dumps those electrons onto pyruvic acid. And then when NADH becomes oxidized or loses those electrons, it becomes NAD plus again, which then goes back to glycolysis, allowing for glycolysis to continue, so then we can make our ATP. Because again, fermenters only make ATP via glycolysis. Right? And it allows us to regenerate our NAD plus. This big arrow here involves different enzymes, and the type of fermentation is all about the enzymes involved, right? So lactic acid fermentation ends up producing lactic acid. Alcohol fermentation produces ethanol. It's all similar in the sense that the enzymes dump electrons back onto pyruvic acid, right? But the different enzymes produce different end products. And some of the more common industrial ones are, at least the good stuff, are ethanol fermentation that produces our yummy, goodney beer and wine. Lactic acid produces some nice foods that we eat. Swiss cheese is propionic acid, right? Those little holes are little acid bubbles that formed during propionic acid fermentation. It's the carbon dioxide, that formed, gets trapped in there. And again, the fermentations are named after the end product. So this will be ethanol fermentation, acetic acid fermentation, and lactic acid fermentation, because they produce ethanol, acetic acid, and lactic acid. Just a little bit about chemotherapy. Right? Remember, we're going to try to revisit this idea of chemotherapy periodically throughout our semester. And I want to talk a little bit about the cyanides. And the cyanides are very highly toxic molecules. And what they end up doing is they inhibit cytochrome C oxidase, which is an enzyme in the electron transport chain. Oops, I should say in the electron transport chain. And by inhibiting this enzyme, essentially now we disrupt the flow of electrons, right? So if we diagram our electron transport chain again, we have our enzymes, right? So electrons get pushed from one to the next. Now, cyanide blocks that, and this prevents oxygen from taking that last electron. So now this one builds up here, the electrons build up, electron builds up, electron builds up, NADH now cannot no longer drop off those electrons, and then everything backs up, right? And this is like the worst traffic accident ever. Everything backs up, no flow of electrons, so cell cannot use the oxygen, cannot produce ATP. Ultimately, the cell dies. Now, fermenters that do not use the Krebs cycle in the electron transport chain, they're not affected by cyanide. Cyanide only works on organisms that respire. Right? So for us, highly toxic. For something like the fermenters, like yeasts, they can survive because they can survive on fermentation. Now this part, we're going to go off the grid a little bit. This is not in your notes, but I thought it was kind of interesting stuff, and maybe an interesting way to help tie in and maybe help make the connections between metabolism, catabolism, and what we just talked about, right? And we're going to talk about our muscle cells. We're going to talk about exercise. As muscle cells, they have a huge ATP demand, right? But there's only a small supply of ATP floating around in the muscles. Only about 3 seconds worth of activity. Just enough to get going, right? So everyone has 3 seconds of activity. And so if someone says, can you grab that for me? You say, I can't do that. That's not true. If it only takes you 3 seconds to grab a pen for somebody, everyone has 3 seconds of activity. After those pool of ATP that's floating around disappears, then your muscles need to create ATP. And there's 3 ways that happens, and that's called the phosphagen system, the glycogen lactic acid system, and aerobic respiration. And these things will all kind of draw on what we just talked about. The phosphagen system is highlighted by this molecule we call creatine phosphate. And if you're a bodybuilder, you maybe take these creatine phosphate supplements. Creatine phosphate essentially is a phosphate-containing molecule. There's an enzyme in your muscles called creatine kinase that transfers phosphate from creatine phosphate to ADP to make ATP, substrate-level phosphorylation. And remember, when we talked about substrate-level phosphorylation, we said it's very fast and easy, but it's not all that efficient, right? So we can make that energy very quickly, but it gets used up very fast, and we don't make really a whole bunch of ATP, right? And you have about 10 seconds worth of creatine phosphate in your body, which is why weightlifters who supplement themselves with creatine phosphate try to get maybe 15 seconds worth or 20 seconds worth, just a little bit more. And as far as for energy purposes or for exercise purposes, the phosphagen system is very important for sprinters or weightlifters who do very low repetition by high weight exercise. Just crank them out, just pound as much weight as you possibly can for a short period of time, because you only get about 10 seconds worth, right? And so their diet is all about trying to increase the amount of creatine phosphate you have, just so you can get that extra 1 or 2 seconds, right? And we all know Olympic sprinters, they only need 10 seconds worth, and then they're done. After that creatine phosphate store runs out, we need a little bit more energy. And that's where the glycogen lactic acid system comes in. Glycogen is a glucose polymer stored in our muscle cells. It's simply strings of glucose together. And when the phosphagen system or the creatine phosphate starts to run out and is gone, the cell then hydrolyzes glycogen. Right? When I say hydrolyze, what are you thinking? That's right, we're taking our polymer, our glycogen polymer, breaking it up into glucose monomers. And then it takes those glucose monomers and then produces ATP via lactic acid fermentation. Fermentation, we just talked about that. Very short process. We saw those 2 big arrows, glycolysis and then lactic acid fermentation. It's slower than phosphagen system, right? It's about 12 reactions maybe rather than 1 reaction, but it can produce more ATP than the phosphagen system. Right? So we have about 90 seconds of energy using the glycogen lactic acid system. Now the problem with this system is that it produces lactic acid, which is an acid, right? So that acid starts to build up in your muscles, and that burning that you feel, that's lactic acid buildup, and it happens after, well, depending on how much glycogen we have and how good our muscles are, it can happen after about a minute and about 90 seconds, and then eventually you have to stop, right? And the glycogen lactic acid system is very useful for kind of mid-range stuff that lasts you maybe 1 to 2 minutes of activity. So swimmers, they're kind of very much dependent on the glycogen lactic acid system. Medium distance sprints, and so for them, their diet is all about carbo loading, getting that level of glycogen up in your muscles as much as possible. Finally, we have aerobic respiration, right? And this is now using oxygen to burn glucose, and you can burn whatever glycogen is left over, because usually the glycogen phosphate system, the glycogen lactic acid system doesn't always use up all the glycogen. Also uses whatever glucose is present in the bloodstream, and then it goes on to your fatty acid reserves, and in very rare conditions like starvation, then it will use up proteins, and it'll do it in this order: any glycogen left over first, then glucose, then fatty acid, and then proteins. Now it's much slower than the phosphagen and the glycogen systems, right? Because of the whole building the damn thing, right? That takes forever to get going. It takes about 2 minutes to really start getting good ATP from aerobic respiration. But as long as you have lots of glucose, as long as you have oxygen, you can supply ATP for hours, right? So that's how people can run marathons, and they run it, they don't walk it, right? Any high endurance activities where you require lots and lots of endurance, it's all very much reliant on our aerobic respiration. So now I hope this kind of ties in what we talked about, our catabolism, equating to energy demands, ATP production. If it didn't, I hope at least it was interesting. Now let's talk about a different type of metabolism or catabolism called photosynthesis. And the term photosynthesis comes from the word photo, which is the conversion of light energy into chemical energy, basically using light to make ATP. And then synthesis, meaning we're going to make organic carbon molecules from an inorganic carbon molecules. We call that fixing carbon. Right? That's taking inorganic and producing organic. And we split these off into two reactions. The photo part is the light-dependent reaction, sometimes called the light reactions. The synthesis part is the light-independent, sometimes called the dark reactions. Right? And we'll kind of look at these two separately. The light reactions happen because of this molecule called chlorophyll. Chlorophyll is usually green. Right? This is what gives the green plants their greenness, same with the algae. But there are other types of chlorophyll. And chlorophyll is a molecule that can absorb light energy. And not only can it absorb light energy, it can pass that energy onto electrons. Right? So when light energizes or hits chlorophyll, chlorophyll grabs that energy and puts it into electrons and charges them up. Makes them high energy electrons. Right? So chlorophyll gains high energy electrons by taking the energy in light and charging them up. And there's two types of light reactions called cyclic and non-cyclic, or anoxygenic and oxygenic. Basically, one produces oxygen, one does not. And we'll look at both. Again, big picture, right? And this time, we're really going to avoid some of the nitty-gritty details. Cyclic photophosphorylation is probably the easiest to understand. It's anoxygenic, right? Does not produce oxygen. And it works something like this. We start off with chlorophyll and light hits it. And when light hits chlorophyll, chlorophyll grabs the energy that's in light and excites an electron. And we always like to draw it like this. You know, we have electron down here, and then boom, now we have this high energy electron. Gets this high energy level. It doesn't actually go anywhere. It just simply has gained energy. Now we're going to use that energy. Chlorophyll passes those electrons to an electron transport chain. What? Yes, that exact same electron chain transport chain we've talked about. So it's a series of electron carrier enzymes in the membrane. Every time the electron gets passed, what happened to my pen? La 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 la. Every time the electrons get passed, what happens? Protons get pumped out, chemiosmotic gradient. Ultimately, and ATP is generated by allowing the protons to come through the ATP synthase, creating ATP, chemiosmosis, right? This is now photophosphorylation. Just like oxidative phosphorylation, building a dam. But we're using the light energy to build a dam, not food energy. Yeah? Great. Now these electrons ultimately return back to chlorophyll, right? Completing the cycle. Cyclic photophosphorylation. Get it? All right. So my question to you now is, how many products do I have? The answer is 1. ATP, cyclic photophosphorylation, produces 1 product. Second question, what is my source of electrons here? The answer is chlorophyll. That's where the electrons come from, right? So my source of electrons is chlorophyll. Third question, what is my final electron carrier here? The answer is also chlorophyll. It's the last thing to touch the electron. Right here now, chlorophyll is receiving that electron. Now it's going to get energized. That electron is going to get bumped up again, and boom. And we're going to continue to do this chemiosmosis thing. Non-cyclic photophosphorylation is oxygenic because oxygen is produced. And you're going to see photosystem 1, photosystem 2. When you see photosystem 1, photosystem 2, just kind of think chlorophyll. And it kind of hits it twice. We're going to kind of ignore that part a little bit. And we'll just start like this. Light excites our electrons. Again, in chlorophyll, light bumps up our low energy electrons, makes some high energy electrons, have our excited electrons. Just like before, those electrons get passed on to the electron transport chain. Now I'm going to talk about this fast because we should know, right? Electron transport chain generates our chemiosmotic gradient. chemiosmotic gradient allows ATP production via chemiosmosis using the ATP synthase enzyme. Now, unlike cyclic photophosphorylation, our electrons end up on NADP+. It's an electron carrier. Notice the P. It's not NADH or NAD+. NADP+. NADP+ gets reduced. Reduction is gain. Forms NADPH, and we'll see NADPH very soon. Okay, so now let's say we let this thing go. Electrons bumped up, NADP+. And electrons bumped up, NADP+. What's missing here? If you said we need to replace those electrons in here, then you're correct. And the electrons here are replaced by electrons from water. So here we have water. Water becomes oxidized because it lost those electrons. Those electrons go to chlorophyll and then allows us to complete our cycle. And then when water gets oxidized, it becomes oxygen, which is our waste product. Right? And that's how plants produce oxygen for us. Also, when you're watering plants, in part, what you're giving them are their electrons. Right? So when you're watering plants next time, you say, here you go, my lovely petunia. Enjoy these lovely electrons that you're being fed. All right, 3 questions again. What is the... Nope. How many products are made? If you said 3, you're correct. ATP, NADPH, oxygen. What is our electron source? If you said water, you are correct. Water is our source of electrons. What is our final electron acceptor? If you said NADP+, you are correct. Right? NADP+ is our final electron acceptor. Gains that electron gets reduced to become NADPH. Very quickly, we'll talk about the light-independent reactions, the dark reactions. It's really only done by those that produce or that use non-cyclic photophosphorylation, not non-cycle photophosphorylation. And it uses that ATP and NADPH to fix carbon dioxide, or take carbon dioxide, which is inorganic, and make it organic. Again, super amount of details. We're really going to get down to the nitty-gritty here. Here's carbon dioxide from our atmosphere. Carbon dioxide now, or the fixing of carbon dioxide, actually requires a whole bunch of energy. You can't just make organic carbon. It requires a lot of energy. 3 ATP, 2 NADPH to fix a carbon. To make glucose, you need 18 ATP, 12 NADPH. So you need a lot of energy just to produce glucose. At the end of the day, we have glyceraldehyde 3-phosphate, which is organic carbon. That gets joined together then to form glucose. So notice now, by the way, in this non-cyclic photophosphorylation, in the Calvin-Benson cycle, we've made glucose. Right? And glucose actually is going to be the food source for plants, for algae that produce or that do this type of photophosphorylation, non-cyclic, and the Calvin-Benson cycle. Their food source is actually glucose, not light. The light is simply used to generate the ATP and the NADPH to fix carbon dioxide. And then they're going to fix the carbon dioxide, make glucose, and eat the glucose. They also have mitochondria. Yeah? Interesting stuff, right? Okay, I've done a lot of talking, but I'm almost done, I swear. All right, so metabolic diversity among organisms is all about classifying organisms based on the food that they need, right? Or their nutritional pattern, their nutritional fingerprint. And specifically, we can look at the basic requirements of life. How do they get their energy? Where do they get their carbon from? And that's basically what we're looking at. There's 2 basic energy sources. And they are light, and if you use, let me circle that in there. If you use light, you are a phototroph. The word troph means to eat, photo means light. And if you use chemicals, then you are a chemotroph, right? So chemotroph use some sort of chemical, whether it's inorganic or organic. You can carbon sources are also come on 2 basic sources. Inorganic carbon, or autotrophs, organic carbon, or heterotrophs. Okay? Now, we can combine these two, one from column A, one from column B, to make an organism with its energy source and its carbon source, right? So, let's combine them. And so we have a photoautotroph, right? So what's its energy source? Photo refers to light, right? So it's light energy source. And then what's its carbon source? Inorganic carbon, or carbon dioxide. How about a photoheterotroph? Again, light energy, right? And its carbon source. In this case, heterotroph refers to organic carbon, right? Photoautotrophs, by the way, are like your algae, your primary producers. They take in light energy, or they use light energy, and they breathe in carbon dioxide, inorganic stuff, to produce organic carbons. Photoheterotrophs are bacteria that can produce ATP using light, but then they still require some sort of organic carbon source to build their biomass. Let's look at chemoautotrophs. So what's their energy source? Some sort of chemical energy. They need to eat something. And their carbon source then is inorganic carbon, or carbon dioxide, right? Some very interesting chemoautotrophs. Rust eaters, for example, they eat rust, a chemical, right? And then they can use carbon dioxide to fix and produce biomass. Chemoheterotrophs use chemical energy, and finally organic carbon. So which one of these are us? If you said chemoheterotroph, that is correct. We are chemoheterotrophs, right? We produce, when we gain our energy from eating food, some sort of chemical, and using organic carbon. Well, finally, we'll get a look at just a closer look at chemoheterotrophs. Chemoheterotrophs are very interesting. They are classified according to their source of organic molecules. Saprophytes, for example, live on dead organic matter. That's us, by the way. Parasites get nutrients from living hosts. Many of our pathogens are parasites. And the metabolic diversity of chemoheterotrophs is very interesting to us because we can use them to provide a lot of potential solutions, but at the same time, they cause us so many problems, right? So Rhodococcus erythropolis, for example, is one that can cause disease in humans and animals, eating off us parasitically, eating our organic carbons, but they can also desulfurize or desulfurylate petroleum, right? So we can actually use that as a method to clean up petroleum waste, right? So if we can exploit or figure out how we can tap the ability of bacteria to eat the bad stuff, we can then perhaps find solutions to clean up some environmental issues. And that was our bioremediation that we talked about. Okay, so we talked about a whole bunch of things this chapter, and it's very important that you can try to conceptualize everything. We talked about one way to do that, by the way, is to draw the pathways in a way that makes sense to you. And if you check the discussion board, hopefully I've posted some videos that maybe show you how you can draw the different pathways, the big picture of each pathway, and try to understand it, get it into your own words, so to speak. If it's not on the discussion board, just send me a quick email. Maybe I forgot, and then I will just simply put it up there for you. Okay, so don't get lost in the details. Look at big picture. By understanding big picture, some of the smaller details should start to fit into place. Have a great module, and I will see you guys next week.